So this lesson is about the A-level topic of cancer. So if a mutation happens in a gene which controls cell division, it can actually cause cancer. And this is because the genes that regulate cell division don't perform as they ought to, which may cause uncontrolled cell growth. So mutations that happen in individual cells after fertilization are called acquired mutations. And if these mutations take place in the genes that control the rate of cell division by mitosis, it can sometimes cause uncontrolled cell division. If a cell divides uncontrollably, the result is a tumour, a mass of abnormal cells. And tumours that enter and destroy surrounding tissue are called cancers. There are two types of gene that regulate cell division. There's tumour suppressor genes and proto-oncogenes. Mutations in these genes may cause cancer. So tumour suppressor genes. When a tumour suppressor gene is working normally, the genes slow cell division by producing proteins that stop cells dividing or cause them to self-destruct, which is called apoptosis. If a mutation occurs in a tumour suppressor gene, the gene will be inactivated and the protein it codes for isn't made and the cells divide uncontrollably because the rate of division increases, causing a tumour. Proto-oncogenes. So when proto-oncogenes are functioning normally, they stimulate cell division by producing proteins that make cells divide. Oncogenes. If a mutation occurs in a proto-oncogene, the gene can become overactive, and this stimulates the cells to divide uncontrollably, resulting in a tumour. And a mutated proto-oncogene is called an oncogene. Tumours and cancers. So tumours can develop for years without any obvious symptoms and can be quite large by the time they're discovered. And not all tumours are cancerous. There are two different types. There's malignant tumours. This is where the cells are cancerous and can spread to other tissues and organs. And then there's benign tumours where the cells are not cancerous and they won't spread. So malignant tumours are cancers. They usually grow quickly and attack and destroy surrounding tissues. Cells can break off the tumours and spread to other parts of the body in the blood or lymphatic system. And then benign tumours are not cancerous. They usually grow slower and are frequently covered in fibrous tissue that stops the cells invading other tissues. Benign tumours are often harmless, but they can cause blockages and put pressure on organs. This would be especially important if this tumour was in the brain because it could press on vital nerves. Some benign tumours can eventually become malignant. So tumour cells. So tumour cells differ from normal cells in quite a lot of ways. So the first one is that the nucleus is bigger and darker than in normal cells. Sometimes the cells can actually have more than one nucleus. They have an uneven or irregular shape. They don't produce all the proteins needed to function properly. They've got different antigens on their surface. They don't respond to growth regulating processes and they divide more regularly than normal cells. So causes of tumour growth. There are a number of different factors that are thought to lead to the growth of tumours. These include abnormal methylation of DNA and increased exposure to oestrogen. So this is for breast cancer now. So abnormal methylation is adding a methyl or CH3 group onto something. And methylation of DNA is an important way of regulating gene expression. It can control if a gene is transcribed and translated or not. And when methylation is happening, usually it plays a vital role in many processes in the body. It's only when it happens too much, which is known as hypermethylation, or too little, which is known as hypomethylation, that it becomes an issue. The growth of tumours can be caused by abnormal methylation of cancer-related genes. So hypermethylation. So when tumour suppressor genes are hypermethylated, the genes are not transcribed, so the proteins they produce to slow cell division aren't made. This means that cells are able to divide uncontrollably by mitosis and tumours can develop. Next one is hypomethylation. Hypermethylation of proto-oncogenes causes them to act as oncogenes, increasing the production of the proteins that encourage cell division. This stimulates the cells to divide uncontrollably, which causes the formation of tumours. So now looking at the role of oestrogen in breast cancer, some women are exposed to more oestrogen than others. And the greater exposure to oestrogen may be because of beginning menstruation earlier than usual, starting the menopause later than usual, taking oestrogen-containing drugs like hormone replacement therapy or HRT. So increased exposure to oestrogen over a long period is thought to rise a woman's risk of developing breast cancer. The exact reason behind this aren't completely understood, but there are a few theories. So it could be that oestrogen can stimulate certain breast cells to divide and replicate, and the fact that more cell divisions are taking place naturally increases the chance of a mutation occurring, and so increases the chance of cells becoming cancerous. Estrogen's ability to stimulate division could also mean that if cells do become cancerous, their rapid replication could be further helped by estrogen, which helps the tumour to form quickly. Other research suggests that estrogen is actually able to introduce mutations directly into the DNA of certain breast cells, again increasing the chance of these cells becoming cancerous. So that is an introduction to how cancer occurs.